So let's move on from a bit more technology side into the biomarkers. So as the title says, we are focused on early diagnostics, mainly cancer, but also this kind of a technology platform or assay platform is also applicable to other diseases. Let's first have, so to speak, the lay of the land. So where you have the disease, so what's the disease progression or the timeline starting from when you don't have any disease to the asymptomatic phase, to the symptoms and the time of diagnostics, and to the treatment and the monitoring. So each of these different phases of the disease actually have different types of biomarkers. And depending on what you want to see or what you want to find, it also determines on how the disease is treated. So what we are interested in is in early disease diagnosis. So everything that happens as soon as possible when the disease is onset on the patient. And from the treatment perspective, what we want to do is push the boundary when the diagnosis happens to do the diagnosis as fast as possible, because the earlier we find the disease, the better are the treatment options or the outcome of the treatment for the patients. So if we want to see this from a different perspective, from technology, on the other side, other end of the spectrum, you have these personalized diagnostics. So you have a lot of more diagnostic options in there based on imaging, on mass spec. So you can do the tumor profiling based on the DNA and on the driver mutations. So you can select different types of diagnostic or use different kinds of diagnostic technologies to actually pinpoint what kind of treatment you have. But that's done on individual patient level. On the other side, you have the population screening. So more patients or more individuals that you have to find. And also because the disease is in an earlier phase, the amount of biomarkers that you actually have available in the circulation or in the biofluids is really small. So this is more of finding the needle from the haystack, and also the technologies available that you can actually use where you have to process hundreds or thousands of samples are quite limited, are basically only for those that are really cost efficient and are applicable for high throughput screening. So there are different types of biomarkers. What we all have been interested in recent years are glycosylations. So I think most of you know what are glycosylations, but just for those who do not, they are basically sugars or carbohydrates that are attached both on the cell surface, but also to the proteins that are secreted outside of the cells. In cancer, it has been known since 1940s that glycosylations change. So starting from the healthy cells all the way to the metastasis of the cancer, you see different kinds of glycosylation changes, and the further the disease progresses, the higher incidence of these aberrant glycosylation is. The other factor that's interesting, or in our perspective, is that depending on the phase of the disease, you actually have different types of glycosylations. So in the early phases of the disease, you have these truncations happening to the glycosylations, and later on, when the diseases start to disseminate, you have, in addition to the tumor cells that are secreted to the circulation, you also have glycosylation changes happening in there. So obviously for us, this is very interesting. If we could target these specific ligands in the early phases of the disease, this would be good biomarkers, especially in cancer. And this can be also seen in immunofluorescence. On the left side, you have healthy pancreas, where you don't have any of the glycans. In this case, it's actually sialylated Lewis X antigen. And on the right side, you have a pancreatic cancer tissue, where on the red, you can actually see that these glycans are there. So they are basically absent in healthy tissue, and they start to form the more advanced the cancer is. So glycans obviously are not the only biomarkers that are available. There are also proteins and lipids, so all kinds of phenotypic markers that are on the outside of the cells, and also within the cells you have DNA and RNA. So try to rationale why we are interested in glycans. I will use a simple analogy. So if you compare, think about these as trees, where you have everything that's within the cell, or the stuff happening below the surface, so the roots, and everything above the surface, the lipids, the proteins, and the glycans are the phenotypic markers. So the earlier in the disease or the changes happening on the metabolic level are more evident the higher you go in here. So when you have disease onset early on, you can actually see the changes the higher you go in here. So even the smallest metabolic changes that are not necessarily visible within the cell, you can actually see it on the phenotype. And the glycans are so abundant on the cell surface and on the proteins that even these subtle small changes actually come to show. And this is something that actually, this was started already over 10 years ago by Emeritus Professor Kim Pettersson. They had this very simple and elegant idea together with the colleagues in Netherlands to take conventional tumor markers, for example, prostate-specific antigen such as PSA or CA125, which is used in the diagnosis of ovarian cancer. And instead of, so to speak, conventional immunoassay, where you actually detect CA125 with the same antibody on the proteins, you detect a glycovariant of that same tumor biomarker. And for example, in CA125 cases, which actually can show you it's elevated in ovarian cancer, but it's also elevated in endometriosis. And that's a huge problem. So if you screen samples, you have same age patients, and you try to find ovarian cancer, and you actually have also endometriosis patients in there, there is no discrimination between endometriosis and ovarian cancer. The healthy ones, you don't have CA125, obviously, because it's not secreted in healthy conditions. But for age-matched patients, you want to find a difference between endometriosis and ovarian cancer. So when you change one of these antibodies in the immunoassay into a one that binds to a glycan epitope, in this case it's STN, you all of a sudden see a big difference between the ovarian cancer and the endometriosis. So very simple solution, and this can be done in very simple way also. And what was okay, there's a zoom in the middle of it. Okay, it doesn't matter. Let's go forward. Okay, nice work. All right. So this same assay actually can be done in a very simple way. So this was published a couple of years ago, and it's done as rapid test. So basically, everybody should know by this time what is a rapid test. So, but this actually uses one of the technologies that Tupi was talking about. So this can be done very fast, directly in the doctor's appointment. And it's not only on ovarian cancer. So this whole glycovariant approach, we have been expanding it in the recent years. So it's breast cancer, colorectal cancer, prostate, bladder, and we are now working on pancreatic cancer. And together with Uniogen, we have been trying to get this out of the university, and hopefully we will see this ultimately in the patient care. So this is one of the examples that we have on these technologies that have been on this translational phase and quite far from the research. Okay, so let's go to the up and coming stuff. So what I have been personally working is things called Xlr Vesicles. So these are basically vesicles that are secreted outside of the cells. There have been huge interest in this, especially as biomarkers, because these tiny information packages that can be also called the social media of cells, because they spread the information within the cells. So they really tell what is happening inside the cells. And they carry all the biomolecular components of the host cell. So from diagnostic point of view, this is obviously very interesting. So if we would have this type of different types of biomarker sets, so the glycosylations, the proteins, and also the DNA and RNA within the lumen of the EVs, and we can actually combine this in the same diagnostic system, then this is really something that can be already, could be called liquid biopsy. So trying to find out what happens in the host cell through these tiny information packages. And also what is very interesting is that obviously for diagnostics is that you can actually find these EVs from all of the biofluids. So they are in the circulation, they are in urine, sweat, tears, wherever. We have a big community who are studying EVs as we speak, and especially for the biomarkers, and trying to also get this to the clinic. There are a number of technological and methodological problems still that we have to overcome before that happens, but there is a lot of active research also in our unit related to these. The first one is, well, it's basically the same thing that we have been doing, the glycovariants, but this time we were trying to look at the transmembrane proteins, and in this case, this is bladder cancer and integrins. So integrins are cell adhesion molecules. They are known to be overexpressed in cancer cells. And so we did this very simple assay again. So we have integrin assay. So we bind it with the same antibody, and you don't see any separation between the healthy, benign, or bladder cancer cases. But again, when you change this time the tracer into a glycan-binding lectin, all of a sudden you ramp up the sensitivity and also the specificity of the assay, and then you can start to see difference between the patient groups. But obviously, this is not something we're going to stop. We want to really combine all of this. So we have the glycans and proteins, and what we have shown that when you combine these two set of biomarkers from two different biological processes, you really see a difference. But ultimately, what you want to see is also the other class of biomarkers, so DNA, RNA, all wrapped up in the same system, and then you really start to see the difference between the patient groups. And then the last one is what's going to happen next is actually it's related to technology. So what has happened, or what started 15 years ago in cell biology, where we moved from the bulk omics or the bulk analysis of cells into the single cell analysis, that has really revolutionized, changed everything. Our understanding on cells is actually happening also for EVs. So we're moving from the bulk analysis into the single EV analysis era. And this is going to also change the whole field again, especially in diagnostics. If you think about that, you can fish out from the bloodstream these different types of EVs coming from the different tissues. And that's really going to be interesting if you can use these as biomarkers in the future. All right, big thanks to the group. We have fantastic cancer diagnostics group, collaborators, affiliations, and of course the funders. Thank you.